Okay, welcome back for the last part of our day. It's a pleasure to reintroduce Dr. Eric Youngstrom, who's going to walk us through this next session with three early career investigators. Come on up, Eric. Everybody fed, watered, feeling like on a pretty good spot on Maslow's hierarchy? Because this is a full day. We've been packing in a lot of information. So am I reading the bios now, Ben? Ben? Yes, please. Yes, please. So we have an excellent set of talks coming up. I'm assuming that they're in the order that they will present. So first we have Nidhi Kulkarni. She's a fourth-year PhD student at UT in the Department of Pharmacology, working under the supervision of who? Ben who? Ben Goldstein. She graduated from Western Ontario with an MSc in Physiology and Pharmacology, and from the University of Guelph with an honors BSc in Biomedical Sciences. My daughters are statistics and chemistry majors, and they would be very impressed. They would say, she's picking real sciences. The Nidhi's research focuses on familial and molecular genetic factors underlying the vascular bipolar link. Thank you so much. Looking forward to hearing your thoughts.
Thank you, Dr. Youngstrom, for that lovely introduction. Hello, everyone. Hope you all had a nice break. My name is Nidhi, as Dr. Youngstrom mentioned, and I'll be kicking off the set of rapid presentations by discussing my work on the association of polygenic risk for coronary artery disease with gray matter structure in youth bipolar disorder. So as many of you may know already, bipolar disorder has been associated with an increased risk for and an early onset of cardiovascular disease. Indeed, the mean age of individuals who develop cardiovascular disease is up to 17 years earlier for those with bipolar disorder as compared to the general population. This enhanced cardiovascular burden is often in excess of side effects from psychotropic medications, adverse lifestyle, as well as traditional cardiovascular risk factors. So coronary artery disease is the most common type of heart disease and the leading cause of death worldwide. Furthermore, adults with bipolar disorder have a greater prevalence of coronary artery disease, and at least double the mortality due to coronary artery disease as compared to the general population. Furthermore, there is preliminary evidence of a genetic overlap between bipolar disorder and cardiovascular disease. Indeed, a recent 2021 paper developed polygenic risk scores, or PRS, for coronary artery disease in more than 61,000 adults with and without bipolar disorder. So as you've heard from Dr. Clemens Zai, a polygenic risk score is an aggregate index of an individual's genetic liability to a disease. So what that essentially means is that the greater number of genetic risk alleles with a large effect size that an individual carries, the higher their polygenic risk score, and thus the greater their genetic risk for that disease. So what this 2021 paper found was that bipolar disorder was associated with an increased polygenic risk for coronary artery disease, even after controlling for the coronary artery disease phenotype in these adults. Several studies have also examined coronary artery disease in relation to the brain and have demonstrated that coronary artery disease is associated with lower total brain volume as well as lower regional gray matter metrics. However, there have been no recent or related studies in a psychiatric population or in a youth population. There is evidence that polygenic risk for coronary artery disease is also associated with the brain, with a recent paper demonstrating that a higher coronary artery disease PRS was associated with greater whole brain volume atrophy, as well as greater white matter hyperintensities. Again, this study only focused on older adults with mild cognitive impairment and Alzheimer's disease, respectively. So given the paucity of youth studies on this topic, we decided to examine the association of cardiovascular PRS, specifically for coronary artery disease, with brain structure in relation to youth BD. We hypothesized that a higher coronary artery disease PRS would be significantly associated with lower gray matter metrics in a combined sample of youth with and without bipolar disorder, and that this association would be stronger in youth with bipolar disorder as compared to the healthy controls. 111 youth were included in the study, including 66 with bipolar disorder and 45 healthy controls. Coronary artery disease PRS was calculated using PRS-CS Auto. Which is a method that estimates the posterior effect sizes for these genetic risk alleles, or SNPs, that were derived from a genome-wide association summary statistic for coronary artery disease cases and controls. Flink was used to weigh the SNPs by their effect sizes, and the adjusted SNPs were then summed to create a polygenic risk score for each of the individuals within our cohort. Brain structure images were obtained using 3 Tesla Philips scanner and processed using FreeSurfer. For our primary analyses, we looked at whole brain cortical volume, thickness, and surface area, and for our secondary analyses, we looked at 2 specific regions of interest, the anterior cingulate cortex and the hippocampus. Vertex-wise analyses was performed using FreeSurfer to examine association between coronary artery disease PRS and whole brain gray matter structure. Linear regression was used for these secondary ROI analyses. All analyses were conducted within the overall sample and separately within the bipolar group and the healthy controls. Covariates included age, sex, the first 2 principal components, and intracranial volume when assessing cortical volume and surface area. Here we have a breakdown of the participants' demographics, which demonstrates that the bipolar group had a greater proportion of females and a greater BMI as compared to the healthy controls. So this slide will demonstrate my findings for the whole brain vertex-wise analyses. And starting with our whole group results, we can see that higher coronary artery disease PRS was associated with lower right inferior temporal volume and the overall sample. Similarly, in the bipolar group, higher coronary artery disease PRS, was associated with lower regional cortical thickness in 5 different clusters. Finally, in the healthy controls, higher coronary artery disease PRS was associated with lower left superior frontal area. Our sex stratified analyses revealed that the findings within the bipolar group remained significant in the females, but not in males. Furthermore, there were no significant sex differences in the combined sample or the healthy controls, only within the bipolar group. In regards to our ROI analyses, we found that higher coronary artery disease PRS was nominally associated with lower anterior cingulate cortex thickness within the bipolar group. However, this did not remain significant after correcting for multiple comparisons. In summary, we found that increased coronary artery disease PRS was associated with lower regional gray matter structure in youth. These findings indicate early changes in neurostructure in relation to cardiovascular genetics, even in individuals that haven't yet presented with clinical symptoms of coronary artery disease. The findings were more pronounced in the bipolar group, suggesting that a higher genetic risk for coronary artery disease is especially detrimental to the brain of those with bipolar disorder. And finally, the findings were pronounced in females with bipolar disorder. Now, studies have demonstrated sex differences in the vascular bipolar link, with females having a 3-fold stronger association of bipolar disorder with coronary artery disease as compared to males. So our findings highlight that this may be due to the increased genetic burden of coronary artery disease on the brain in females with bipolar disorder, and suggests the role of sex in the underlying mechanism. There are certain limitations that should be addressed. Firstly, given that this was a cross-sectional study with data points taken at a single time point, our findings do preclude any causal inferences or conclusions regarding the timing of the observed results. Secondly, the sample size did not provide adequate power to evaluate small effect sizes. And lastly, we did not account for environmental or epigenetic factors that could potentially affect our findings. Moving forward, future longitudinal studies are needed to examine the association of coronary artery disease PRS with neurodevelopmental changes over time, and to discern mechanisms underlying the observed sex-specific findings. Continued research on this topic has the potential to change the conceptualization of heart-brain associations among youth with bipolar disorder, and findings may ultimately inform clinical practice by demonstrating the value of optimizing cardiovascular health for neuroprotective health in high-risk youth, and may also help reduce stigma associated with psychiatric illness. Before I end off, I would like to thank all patients, participants, their families, and funders for making this research possible, and of course to Dr. Goldstein and the rest of the CYBD research team for their continued support in my work. Thank you.
How many of you is this the first time at a scientific conference? A couple of you. We're on a streak where all the speakers are ending on time. That never happens. So well done. Lovely presentation, and Dr. Phillips.
So thank you, very nice findings. I was really intrigued by the whole of the brain matter findings. And I'm just wondering, would you mind putting up the slide, if that's possible, showing all the different findings?
Here.
Yep. And you know, I was just struck by the most significant finding that you're showing there on the left-hand side, because that looks as if the higher the PRS score, the lower the grey matter in that left part of the QRS, which is the left ventrolateral prefrontal cortex. So, you know, I'm just, can you say a little bit more about, you know, what all that means in terms of the big picture? I mean, how you conceptualizing that? Because that, to me, is so important. Cardiovascular risk with the region that I'm interested in.
Right. So, great point. So, like you've seen, and multiple studies have shown all the regions implicated here. So for those who can't see it, the pars superocularis, the rostral middle frontal gyrus, the insula, which I know someone here was very into, and suprafrontal gyrus. So all of these have been implicated in bipolar disorder specifically, especially for mood regulation, right? So considering that it is coronary artery disease, genetics have specifically mapped onto these regions, kind of give credence to the fact that maybe what is driving this bipolar disorder or this mood dysregulation may not just be like a purely psychiatric issue, but it might have like an underlying causal cardiovascular, cardiovascular.
It does speak to the interface.
Yeah.
Cardiovascular factors and neural circuitry, which is a very important point to make.
100%. And I guess continuing off of that, we say cardiovascular risk factors, and we did control for cardiovascular risk factors specifically here to see if that could be a potential thing that was driving this change. And the results remain significant even after controlling for the risk factors. So this seems to be going beyond just the effects of cardiovascular risk factors and seems to be kind of honed in on the fact that it's a genetic driver. Yeah.
And I guess the related question is, which specific genes are in that PRS? That's a big question. That's a big question.
Yeah. So the question was, sorry, I should repeat the question. The question was which specific genes might be related, might be driving this. So unfortunately, we cannot really detect exactly which genes in a PRS are driving it, because the whole point of the PRS is like they're all genes with a very small individual effect size, but when you combine it, you have the positive effect. Yeah.
What the flavor of those genes are in the PRS score, but that's we can talk about.
Yeah, for sure. Okay. No worries, thank you.
So Dr. McIntosh and then Gary.
I was just dumbing it down, higher PRS is bad.
It seems to be detrimental to the brain, yes.
But no, like a higher score.
Oh, yes. So a higher score would theoretically indicates that you're at a genetic risk for that disease or phenotype. It doesn't always have to be disease, but in this case, it's coronary disease. Yeah.
Now, well, is there any hypothesis why it's so dramatically different for females? And is there studies that have a much larger sample size? Can you really, like, I don't think you have a very large sample size.
No, that is a very astute observation. We do not. And that's another thing. We need larger sample sizes to run the study. The stat that I mentioned where females have a higher association of bipolar disorder to coronary artery disease, that was done in a UK biobank sample. So that's thousands of people. But in terms of the cardiovascular genetic link in females versus males for bipolar disorder, that's not being done the way this has here. So that's definitely something that needs
Well,
to be.
can you suggest something like, could it be environmental?
Oh, I see what you mean. Oh, so we're specifically looking at genetics, and sure, like epigenetics could play a role. So that was a limitation of the study where epigenetics can theoretically affect your genes and thus can have an effect on our findings. I'm thinking maybe it's more so it could be hormones. So there have been studies showing the hormones or oxidative stress that might be different, or inflammation that might be different.
Hormone?
Estrogen. Yeah, estrogen is higher. It's also, it would be interesting to see even, changes during the monthly phase, like hormonal changes during the month, how that might affect it. But yeah, it's up in the air, what could be driving it. Yeah.
Are there regions of the brain that don't show the difference? Just looking for when you first showed that, I was struck by the consistency
Mhm.
of the finding across different regions. Is it a sort of, this is affecting the whole brain? Or are there regions that you could look at that don't show?
So this was the way the vertex-wise analyses kind of takes place is we only really saw the regions that passed the cluster-wide p-value. So these are the results that came out of this analysis, and I'm showing all the results that we got for these analyses, and they all were in the same direction.
And so everything else that is not shown, we would presume is flat.
Flat, or it theoretically could be in a positive direction, but it wasn't significant, so it didn't show through.
Mhm.
Yeah.
Would we see if there's anything in the chat, or do you?
Oh. Okay. Okay.
What funding were you happiest to see?
I was, well, happy is, I guess, subjective, but it was very interesting to see exactly how robust the finding was in the bipolar brain as compared to the combined and healthy controls. I was expecting, I honestly didn't go with any expectations other than we're probably going to hopefully going to find a negative association based on literature. Again, this is such a new topic with such very little data on it in the literature. So I was very happy to see how robust the findings were in the bipolar, suggesting, hey, we're actually hitting our mark. Like our theories are kind of starting to make sense. So the cardiovascular disease in bipolar is especially detrimental as compared to the general population,
And if someone was really impressed and was like, I would like to give you $1 million, what would you want to study next?
Ooh, $1 million? Ooh.
Yeah, Florida gives you $1 million, sure.
I hope so. I feel it's definitely something that needs to be studied more, especially in youth. Genetics, especially the deeper you go, the more genes you need to look at, it gets expensive. So deep gene sequencing would be amazing to specifically parse out every single gene in a person, then put it into the score and then see which genes kind of even pop out and might be associated more so with the brain. That would be great. So doing some deep genome sequencing, I know that costs $1,000 per person. So $1 million could go a long way with that. Yeah, I would love to do that. And maybe also more scans and more longitudinal scans would be amazing again for that, to see how a higher PRS affects a youth over the lifespan and starting young, if this is already kind of present when they're babies, or when does this start to present, in the brain.
Now, one more question, your sample, what percentage was those who were hospitalized and those which are out in the general community? Like, does this apply to people with non-current symptoms?
So these are all, so this is a clinical sample. So these are all through our registry. So they've come in and been referred to Ben and have been diagnosed with bipolar disorder. There are some that are maybe going through manic symptoms or depressive symptoms. It's a mix of that, but the bipolar group is a clinical sample, and the controls were garnered from the population.
Okay, so you're talking about people who were actually admitted.
Or referred to Ben, yes,
Yeah.
for bipolar disorder.
That is inpatients or
So they were they're inpatients and being treated by Ben, so they are being treated for bipolar disorder.
okay, well, I was wondering if that had any, like, you may get more women coming in.
So make it as in like specifically have more women come in?
Well, I like to understand why it's significant heart risk for women. Like, men have a heart risk too.
100%. I think most people especially in the general population, I think men in general trend to have a higher cardiovascular disease risk and mortality than females. It might decrease a bit as the females get older and don't have that protective estrogen, I guess. It's interesting here. That's something we need to look into more. Why specifically females with mental health? It's not just bipolar disorder seem to have a higher cardiovascular disease risk. For example, those with depression also do. It might be, like you said, due to societal limitations, where females don't necessarily always receive the same amount of treatment or care as compared to males, because they do present differently with symptoms for cardiovascular disease. This case, this was specifically just looking at genetics. So again, that shows that maybe while there is a higher cardiovascular disease risk in female with mental illness, it's not just due to these societal discrepancies, but there might be a genetic driver like hormones that might be playing a role. But that's something we need to look into. Yeah, no worries. Yeah.
All right, that was wonderful and a very, very lively question and answer as well. So without further ado, it is my pleasure to introduce our second panelist, Kassandra Zachos, who's a fifth-year student working on her PhD under the supervision of Anna Andreasa, who is, there you are. So she's focused on the involvement of the mitochondria and the relationship between mood disorders and metabolic syndrome. So we've all been primed for that with Dr. Phillips' talk as well. Looking at uncovering the relationship of mitochondrial blood-based biomarkers to improve the understanding of the mechanisms underlying the chronic diseases. Her work incorporates nutritional implications. This is so on point after the talks this morning, and these multifactorial diseases. So without further ado, a warm welcome.
Hi, everyone. Thank you for having me today. And as I was just going to say that, everyone that's spoken today has set me up perfectly for this talk. So I'll take you through a little bit about what I've been doing throughout my PhD. And it is focused on mitochondrial biomarkers and metabolic syndrome in bipolar disorder. If you know anything about the Andreasa lab, we love the mitochondria. Mitochondria is the powerhouse of the cell, but it does so much more than that. It is a signaling and integration hub, and it is necessary to regulate homeostasis throughout the entire body. we noticed that in many chronic diseases, the mitochondria is likely playing a role, as in a lot of these diseases, the organs that are implicated are organs that have very high energy demands. Specifically in bipolar disorder and in my work, we focus on the brain and mitochondria. This is really important as the brain requires up to 20% of our total energy budget. That's a ton of energy that we consume day in and day out just by one organ in the body. Specifically, there is a high mitochondrial density in the synapses, and that is to establish membrane excitability and to execute complex neurotransmission and neuroplasticity functions. I don't need to tell you all today what bipolar disorder is. It's been made very clear and highlighted where we stand in the literature and how much further we have to do to understand this complex chronic disease. What we want to do today with our work is shift a little bit of the thinking that bipolar disorder is not just a psychiatric disease, it is a metabolic disease. And one thing I do want to highlight is just how complex and heterogeneous bipolar disorder is. And we think that may be in part due to the mitochondria. Mitochondria are incredibly complex organelles. I'll get into a little bit of their own heterogeneity and their mitochondrial DNA shortly, but I want to piece together a few really big picture things and why they might actually fit together in a really large puzzle. A lot of individuals with bipolar disorder actually experience metabolic syndrome. Those with bipolar disorder have a doubled increased risk for metabolic syndrome, and it ultimately unlocks this cardiovascular vulnerability that we've all just heard about. Unchecked metabolism ultimately leads to inadequate and ineffective treatments in these patients. In fact, individuals with both bipolar disorder and metabolic syndrome, of this population, over 2/3 are not adequately treated for their metabolic consequences. This leads into this feedback cycle where we're worsening the bipolar disorder symptoms or exacerbating them, which in turn exacerbates the mitochondrial dysfunction and the metabolic syndrome. And it's an ongoing process that we really want to try and intervene. As I mentioned, mitochondria, in many eyes, specifically ours, is the core of metabolism. And we can get dysfunctional mitochondria for a vast number of reasons. I'm not going to take you through all of those today. We'd be here all day. But two of the reasons why we have dysfunctional mitochondria I will highlight today, and they drive our research. The first is metabolic shifts, and this occurs due to dysfunctional mitochondria that could be due to damage or stress, and ultimately we shift from aerobic respiration through the mitochondria and oxidative phosphorylation, but we shift to anaerobic respiration, and we primarily produce energy through glycolysis. This leads to a buildup of lactate. Lactate in bipolar disorder is something that we know are related. We don't have a great understanding as to why, but there has been a good amount of research to highlight that lactate is a concern in bipolar disorder patients. High lactate leads to increased gluconeogenesis, increased lipid metabolism, increased cholesterol metabolism, and glutamine metabolism. Lactate is a known biomarker in other diseases, but it hasn't been thoroughly investigated in the context of bipolar disorder. The second biomarker that I'll walk you through stems from mitochondrial DNA. The mitochondria has their own unique genome, and it is maternally inherited. Mitochondria, due to its shape, it's very different from the nuclear DNA. One, it's a lot smaller. Two, it has a circular plasmid-like shape, which makes it more susceptible to breakages, fragmentation, and copying errors. For this reason, in a state of stress, we can have perturbations and fragmentation in the mitochondrial DNA. Another important factor is mitochondrial DNA is heteroplasmic. That essentially means that we can have different levels of mutations in different mitochondrial DNAs throughout the entire body. So in the muscle, we might have a mutation rate of 60% compared to the brain, which might be 95%, compared to the blood, which might be 11%. So in different organs, our mitochondria functions differently, and that could help us understand the heterogeneity we see in bipolar disorder. As I mentioned, when we have stress or trauma, the mitochondrial DNA fragments, and this gets released into the periphery, and is called circulating cell-free mitochondrial DNA, or CCF-mtDNA. When it's in the periphery, it acts as a damage-associated molecular pathogen and can ultimately lead to downstream consequences, such as pro-inflammatory gene expression and the formation and activation of the NLRP3 inflammasome, which ultimately leads to low-grade chronic inflammation that we see in some bipolar disorder patients. So the objective of our work is to test whether mitochondrial metabolites, specifically lactate and CCF mtDNA, are associated markers of metabolic syndrome in bipolar disorder. We hypothesize that lactate will be associated with metabolic syndrome and that CCF mtDNA will be associated with higher levels of C-reactive protein, which is a marker of acute inflammation. So for this study, we worked with our collaborators out in France, and we specifically the Fondamental Foundation, and this is run by Dr. Marianne Leboyer. We have two cohorts that I will be talking about. The first is called FACE-BD, and the FACE-BD cohort consists of over 800 patients with stable bipolar disorder. Then in our second cohort, which is a replication or validation cohort, which is a little bit smaller, it includes patients that have stable BD, acute BD. We also have non-disease controls and individuals with acute schizophrenia. So I want to take you through a little bit of the demographics. I do want to highlight the age. This did not specifically focus on youth bipolar disorder, but we do have participants as young as 16. This is also a snapshot in time, and this is not longitudinal data, but we do plan on investigating that later on. So the first of our findings, we essentially collected this plasma. We ran a colorimetric assay to determine lactate, and then we did a DNA extraction and TACMAN qPCR to determine CCF mtDNA copy number. And once we did those relatively simple assays on all of these patient samples, we were able to conclude that lactate did associate with metabolic syndrome, which you can see in the figure on your right. So higher plasma lactate concentrations were associated more positively to metabolic syndrome. Lactate was also associated to higher levels of triglycerides and glucose. CCF mtDNA was associated to CRP and not metabolic syndrome. So then we wanted to continue this study, and to drive the rest of our thinking, we primarily switched over to lactate, as this study does encompass metabolic syndrome. And we were able to take two subsets of our population, those with very, very high lactate and those with clinically low lactate. We then conducted mitochondrial targeted metabolomics using liquid chromatography mass spec. And essentially in these two populations, which included 50 participants with high lactate and 50 participants with low lactate, there was a subset of metabolic syndrome participants in both groups. We then used mitochondrial targeted metabolomics, and all the metabolites we used are seen on the heat map, to ultimately infer if we can stratify these patients any further using a mitochondrial-based profile. Uniquely, what we see here is you can see that the groups with high lactate actually have similar metabolic profiles compared to individuals with low lactate. Initially, we suspected that perhaps the individuals with metabolic syndrome would have more similar profiles, but that's not the case that we see here. So I think this figure really highlights that we need to reframe our thinking on how we stratify these individuals, and this can help us infer on how to better personalize treatment to then have more efficacious treatment for these individuals and their metabolic syndrome. The figure or the little map below ultimately shows which metabolites were strongly correlated. And what was unique was individuals with high lactate had really high levels of alpha-ketoglutarate and citrate, and they're both responsible in fatty acid production and breakdown. So that was unique and definitely warrants further investigation. Lastly, to incorporate our second cohort, that we looked at the IGIV cohort, which had groups from both the acute bipolar disorder, stable acute schizophrenia, and non-disease controls. You can see the box plot on the red is our FACE-BD cohort, and then when we assess lactate levels compared to the acute participants, both with BD and schizophrenia, they have higher lactate levels compared to stable BD. Both of these groups, or all of these groups rather, had higher lactate levels compared to non-disease controls. So you can see, depending on the disease state, we do have different lactate levels. So this warrants further explanation on how these metabolite profiles then change during these different disease states. with that being said, I wanted to thank our lab for everything that they've done to help me with this work. I want to thank Anna for everything she's done to support me, and for everyone here for having me today. This is just one step in a series of studies that we hope to move forward and conduct. There's a lot of work left to do, but it does offer some promising results to potentially personalize treatment for these individuals and improve their overall quality of life. Thank you.
And you kept the streak alive.
Thank you.
Done on time. So questions? Dr. McIntosh.
Can you help me understand? It's not my area.
Yeah.
I was a little confused about where the circulating mitochondrial DNA. What was the result with respect to the circulating?
Yeah, so circulating, here, you can see it right here. So essentially, we hypothesized that circulating cell-free mitochondrial DNA would correlate to an immune marker. We didn't measure too many immune markers throughout this study, or that with our collaborators, so we weren't given all of that information, whether they've measured it or not. So it correlates with CRP. It did not correlate with metabolic syndrome, which is why I didn't focus on it throughout the rest of our study. We had a feeling it would correlate with an inflammatory marker, but we didn't know how it would associate with metabolic syndrome. It has been elevated in some bipolar disorder patients, but we don't have a good understanding as to where it is.
My real question was, which organ is this circulating mtDNA? Where's it coming from? Is it the brain?
Could be coming from anywhere.
Can it get past the blood-brain barrier?
Yes, it can. Yeah.
So the organ, it could be the brain.
It totally
but
could.
be the muscles.
Yeah. Yeah, it could. And yeah, it depends on how much mitochondrial dysfunction individuals have where. And we don't assess mitochondrial dysfunction or heteroplasmy in any of these individuals. So.
Same
Yeah.
with the lactate.
Sorry?
Does lactate also cross blood-brain barrier?
So we can measure lactate in the brain. It's a lot harder to do. We actually have collaborators, part of the study, that are measuring it in the brain. I just don't have access to that data. And it'll be really interesting to see how in these participants it correlates, because we don't have a great idea that if lactate in the blood corresponds to lactate in the brain.
Neat.
Yeah.
This is a methods question.
Yeah.
Was it at rest? The blood collection was?
Yeah,
at
yeah,
rest?
it was at rest. Yeah, and this is just a baseline plasma collection. Yeah.
Without the,
Yes, it had a tourniquet. Yeah,
it did.
yeah. Sorry.
So, can you say a little bit more about the relationship between lactate, alpha-ketoglutarate?
Yeah.
and
Yeah, that's a great question. So alpha-ketoglutarate is like the rate limiting step in the citric acid cycle. And depending on, glutamine production and citrate production, alpha-ketoglutarate could, if there's excess citrate, it could then feed into the breakdown of, or the increase of fatty acid production. And it could push alpha-ketoglutarate back into citrate, or it could go the opposite direction. So alpha-ketoglutarate is a really interesting finding that came up in the metabolomics that we have some understanding, and there definitely is literature of elevated alpha-ketoglutarate in bipolar disorder patients. Not a ton of understanding as to exactly where it fits in the whole picture, and truth be told, it could be different for everyone. Yeah.
It could be that obviously elevated lactate equals metabolic syndrome.
Yep.
But could there be a compensatory mechanism by
Absolutely.
which your ketone bodies are produced more in compensation for the glycolysis and lactate, trying to push the ketones into the picture a little bit more?
Yeah.
And so having too much lactate may actually conversely have compensatory effects and make metabolism more efficient down the line. And I'm sure that's what Michelle was with that's another story. But
Yeah.
It seems a bit counterintuitive, but what you're saying, there could be a link.
Absolutely. And I would like to say that there probably is. We like to think that if there's any glucose in the picture, we're not going to rely on ketone bodies to produce energy. I don't know that that's necessarily true. I just don't know that we've investigated it enough. Yeah.
Any questions in the chat?
was wondering, I don't know much about mitochondrial function.
Yeah.
either. Why not add the analysis of the basal functional capacity, the Duke activity status index, as a ways to stratify patients as well? You know, lactate is also an index of muscle activity,
Mhm.
so I don't know if you consider this, if it's worth it.
From our background literature and understanding, it wasn't something that we initially wanted to explore, something we could potentially explore down the line. There's a ton of data to work with these collaborators. So we started simple and maybe in the future.
May I just also make a comment about, you know, it is, as you say, possible to image lactate with MRS and.
Yeah.
You know, with
Yeah.
cells in it.
Yeah.
And I think we'll be in a position to relate the cerebral lactate with some of these proof of markers that you're looking at. So
Yeah.
that might help answer this question and getting rid of the muscle produce lactate.
I'd love for us to repeat
Absolutely.
again.
Oh, I'm so sorry, everyone online.
Can you repeat the gist of what Dr.
yeah.
Phillips just said?
Essentially, her lab is also working on MRS and imaging lactate in the brain, and it might help answer some of the questions that we've had about if lactate in the periphery corresponds to lactate production in the brain, as something we don't have a ton of information about just yet.
Could you go back a couple slides? This is also, thank you, Michelle, for
Where would you like me to go?
the heat map.
Oh, yeah.
That one.
Yeah.
Thank you for asking a methods question. As a data science nerd, I'm like so the plot looks like what I've seen with network analyses
exactly.
a lot is.
Yeah, it's just a pathway analyses, and essentially it takes the data from the heat map and you plug it into something called Cytoscape, and it produces this really nice figure that helps us highlight which
Where the
metabolites are connected more
Right.
strongly. And that's depicted by the thicker line.
Yep.
Yeah.
So is this the same data, two plots?
Exactly, yeah.
I was hoping that was the
us
case.
visualize it a little bit differently for people who
So
need a different
now
picture.
audience, get ready for an audience participation moment. If these are two different ways of visualizing the same data, which do you think is easier to understand? How many people prefer this one? Raise your hand. How many people prefer the heat map? That's awfully close.
very
Hands-
close.
hands down one more time. How many prefer the network? How many prefer the heat map? The one thing that I see that the heat map does that this doesn't is tell us the direction of the effect.
Yeah.
Could you color code the lines?
Yeah,
in this?
absolutely.
So if this was more colorful and showed you the direction, then who would prefer the mashup of? Okay.
Good to know.
All right.
Thank you, everyone, for your feedback.
Yes.
For those online, if you prefer the heat map over the network, please let us know.
So, and a heat map that's showing us the direction of the
Yeah.
association as well as the magnitude would be
I appreciate that.
chef's kiss.
Thank you. I'm going to incorporate that in my next committee
Yeah,
meeting.
me too.
Perfect.
So what finding were you happiest about?
Oh, definitely the heat map. Like just this metabolite profile. I don't know that we were fully expecting such a stark difference. So that was really cool. Yeah, it's hard. You go into science, you want to have expectations, but you know you can't. And
Yep.
you kind of have to let the data drive you. And this was a really cool finding that really did that. So yeah.
And you had to wonder if it was common. The $1 million question.
Yes.
What would you invest in?
Oh, that's a great question. So this was done with a population in France. I would love to do something a little closer to home. I would love to be able to do this longitudinally. And so I didn't really highlight youth bipolar disorder in this talk based on the participants I have, I can't do that specifically. But we know that type 2 diabetes, metabolic syndrome, the incidence is happening at a much younger age these days, and would be interesting to follow up participants from adolescence over the span of their life to see if metabolic syndrome does develop alongside their bipolar disorder, if they've had symptoms of this underlying the whole time that we don't know about. So if I could do something like that and do all of these measures over a series of time, that would be really, really cool.
That would be
It
really,
would
really
be.
cool. Is the band Bare Naked Ladies a Canadian band?
Yes.
Yeah, so I've had them singing, if I had $1 million, I would do a longitudinal study.
Yeah. Yeah, and we do have some longitudinal data. I just haven't analyzed it yet.
Fantastic.
Yeah.
All the way.
Yeah. Yeah.
Your thoughts or what you know into that functional.
Yeah, good question. I can't speak specifically to MAOA. What I didn't include in today's slides was also the analysis that we're working on with the medication these participants are taking. One of the most common ones in this cohort is valproate, which we know is toxic to the mitochondria. And there's a bunch of others. Sorry?
Toxic to my.
Yes.
Why do they put?
That's a great question. I think it's weighing the cons and benefits, which I think is a lot of the case in medicine. And I'm not a clinician. I could be entirely wrong in even assuming that. There are benefits to it, so I can't take away from those benefits either. And there's a lot we still don't know about these medications and mitochondrial function. And I think as we learn more, it may change. I can't say for sure. Like I said, I'm not a clinician, so I can't speak to that. But I think that's a great thing to consider, and medication is a really important part of this picture that I don't have here today, as well as diet and lifestyle. And so it's a big picture that encompasses a lot of different things, and we're just trying to narrow in on one small piece. Mm-hmm.
On that point, I'd just like to point out that valproic acid deprecated does boost GABA.
Yeah.
And does to some extent.
Yeah.
Protect it.
Mhm.
But it's when it gets over the top very high, I think that's what we're talking about.
Yeah.
It's toxic.
Yeah.
And it depends on others. And so it's good that this is otherwise nobody would be.
Yes. Thank you for.
But you have to be careful, like this. That's attractive.
Yeah. Thank you for that clarification. Like I said, I can't speak to clinic and all of that.
All right.
Thank
Wonderful
you.
job. Thank you for that too. All right, so rounding out the set, it's my pleasure to introduce Ricki de Koning, a PhD candidate from the Department of Psychiatry in the Amsterdam University Medical Center in the Netherlands. She works under the supervision of Dr. Brenda Pennings and Manon Hilligers. And I've had the privilege of speaking with Manon, working with Manon many times over the years. I hold your team in all these teams in very high regard. She has a background in biology and neuroscience. Her PhD is working on the Mario project, studying the development of children where a parent has bipolar disorder or depression. Today, she presents some of the first findings from the Mario project. Welcome.
Well, thank you for this introduction. So indeed, I am Ricki de Koning. I'm from the University Medical Center of Amsterdam, and I'm very grateful to be here today. And I'm also very excited to be here today because I have some first results of the project of my PhD, the Mario project. Oh yeah, so first some learning objectives, and I don't have anything to disclose. So the first one is that you understand the importance and value of studies with offspring of parents with a mood disorder. And the second one is that you know the differences in prevalence of psychopathology between offspring of parents with a mood disorder versus offspring of healthy control parents. Yeah, so some background first. About a quarter of children grow up with a parent with a mental illness. And having a parent with depression increases the risk for developing, a parent with a mood disorder increases the risk for depression in children by 2 to 3 times. And this is because these children have a double burden for mental illness. On the one hand, they have a genetic vulnerability to develop one. But on the other hand, they also often grow up in a less favorable environment with, for example, social isolation. And here you can see an historical overview of offspring studies. It's a figure by Sandstrom et al. And you can see that in the earlier days, most offspring studies focused on schizophrenia. So that's the red bars. And then later on, more studies had a more cross disorder approach. So they focused also on bipolar disorder. But still, there's not been that much focus on depression, but it's coming more lately. But there are still only 2 recent studies that focus on mood disorder specifically, so bipolar disorder and depression. One of which is here, the Forbo study. So that's where the Mario study comes in, started in 2019, and we focus on mood disorder, so depression and bipolar disorder, and also a little bit on schizophrenia. Yeah, so like I said, we focus on bipolar and depression offspring, which I call high risk in this presentation, and on control offspring, and we also have control offspring, so that's offspring of parents with no mental illness. It's a longitudinal cohort study, and our central aim is to better understand the intergenerational transmission of mood disorders. And we want to do that by finding differences in psychopathology in the high-risk offspring versus control offspring, and within this high-risk offspring to identify risk and resilience factors. So we started in November 2019, and the projected end date is in 2027. And actually recently the design paper of the project was accepted, so this will be published soon. So it's a large Dutch consortium. It's a collaboration between 5 university medical centers in the Netherlands. The PI's are Manon Heligers and Dr. Brenda Pennings. And the project coordinators are Melanie Horsval and Annabel Vreker. We also collaborate with patient associations and knowledge centers. So about the design of the project. So we've just finished baseline inclusions, and we've included 403 offspring of affected parents, so that's the high risk group. And we've included 147 offspring of unaffected parents, so that's the control group, and the age at baseline was between 10 and 25 years old. And we recruited these offspring through their parents who participated in previous studies on mood disorders, which also means that through the data that's already been collected on these parents, we have very extensive phenotype and genetic data of the parent. Yeah, so the assessments are spread over 4 years, and at baseline and 3 years, sorry. And at baseline and 3 years later, they come to the hospital, the offspring and their parents, for an interview. And at all 4 assessments, they fill out online questionnaires. And so one parent participated in a previous study, and we also collect data on the other parent, some questionnaire data and saliva or blood as well. Yeah, I think a lot of you are familiar with the kitty sets, but I'll briefly explain what this is. So it's a semi-structured clinician-administered version of the kitty sets. And with this, we assess past and present symptoms of child psychiatric diagnoses according to the DSM-5. And it consists of 3 components. First, there's just a general unstructured interview about the general mental health of the child. Then there's a screening interview, and if one of the diagnoses is more likely then there is also a supplement to see if the child really fulfills the criteria for this diagnosis. Yeah, and then part of the online questionnaires that the children and the parents fill out are the SIBA questionnaires. Possibly also some of you are familiar with this. So it's a collection of questionnaires with which we can assess maladaptive behavior in the children. So this is this reflects emerging psychopathology. And there are different versions based on the reporter and the age of the child. But the questionnaires are more or less the same in all of these versions. So for the self-report version, we have the YSR and ASR, and YSR is for children between 10 to 17, and the ASR for 18 or older. And then we also have the parent-reported version, so what the parent reports about the child, and then those are called the CBCL and ABCL, so again, for under 18 and over 18. And from all these questions, so it's a large questionnaire with about 120 questions in it, you can calculate an internalizing score, an externalizing score, and a total problem score. And this total problem score is an addition of all questions together. Okay. So then some first results, and I have to warn you, these are preliminary, so these are not like final. We still have to do some data cleaning. Yeah, so first the demographics. So about a quarter of the offspring is high risk, and we have slightly more females than males, and the mean age at inclusion was 17. Yeah, and here you can see the lifetime diagnoses that we found with the KidiSats interview. So we didn't find any differences in bipolar disorder. So the high-risk offspring, yeah, it seems a bit high here, but that difference isn't significant. The high-risk offspring doesn't have bipolar disorder more often. For the depression, they do. So we do find more depression in the high-risk offspring, and we didn't find any differences for psychotic disorder. And this is basically the same picture, but now I split it for parental diagnosis. So for whether the parent has depression or bipolar disorder. Again, for the bipolar disorder, we didn't find any differences, also not between whether the parent had depression or bipolar disorder. For the depression diagnosis, you can see again that the high-risk offspring is more often diagnosed with depression, but between whether the parent has bipolar disorder or depression, we didn't see any differences. And for psychotic disorders, we didn't find any differences between any of the groups. Yeah, this is on comorbidity. So here you can see that the high-risk group more often has at least one diagnosis. And also they more often have multiple diagnoses. Yeah, and these are the results of the SIBA questionnaires. So first, this is the self-report. So for the YSR, so that's that's for the self-report for children between 10 to 17. We didn't see any differences in any of the problems scales, but for the age group 18 to 25 self-report, we did find that they reported more externalizing, more internalizing, and more total problems. And for the parent-reported version of the questionnaire, we actually the same picture emerges. So between the age of for the parent who reported about their children of 10 to 17 years old, we didn't see any differences. But for the parents who report about their adult offspring, we did find more externalizing, internalizing, and total problems. Yeah. So these results suggest, as expected, that there is a higher prevalence of mood disorders and that there are more internalizing, externalizing, and total problems in the high-risk offspring, especially in the adult offspring. And next steps will include to examine the TRIA data. So that means, for example, that I'm going to see how parental clinical factors relate to how high the risk is that the offspring develops psychopathology. So we just finished baseline inclusions, but we still have 3 years to go, at least 3 years for follow-up assessments. And also there are this is only one part of the project. We were also working on the validation of a screening instrument for this high-risk group, and also we're working on testing the efficacy of an online intervention platform for this group. And that was it. I'd like to thank all participants and our funders, ONMW, and also our youth and adult panel who helped with setting up this study. And these are my references.
That was wonderful.
Thank you.
How many languages do you speak?
Oh, well, so I speak Dutch and English, and in high school in the Netherlands, you need to have German and French as well. So, and I did Latin and Greek as well. So, yeah, but I don't speak all of them, but yeah.
Yeah, so I am always impressed when someone delivers really good science in a second language. My amygdala remembers at the International Bipolar meeting in Mexico City, I was a speaker and the chair gets up and addresses the audience in Spanish and says, "This is a wonderful symposium. We are becoming a truly international meeting. Everyone will speak in Spanish." And I'm like, except for Youngstrom, who has the heart of, so I got to do my talk in English and I've never been so grateful.
Yeah, well, we grow up with English, so it's different for me to speak English than it is to speak Spanish for you.
So questions?
Yeah, oh, sorry.
First of all, thank you, congratulations. This is a very impressive study. I worked with Boris Burma on the bias study, and these studies are so hard to do. I mean, just recruiting people, so congratulations. I know what a lot of work they went through. I have a couple of questions really.
Yeah.
I guess my main question is, I was impressed to see that in the youth offspring, there were more disorders, comorbidity. Can you say a little bit more about what those disorders were? Because this gives hints about prodrome for bipolar disorder. I mean, presumably it's more than just depression and ADHD, kind of anxiety. I mean, can you just sort of give
Yeah, so depression was the most common disorder. But next, anxiety indeed. So social anxiety and generalized anxiety was most common. Also OCD and PTSD, but these are two disorders that's one of the things that I think still need cleaning. Yeah, so, and also ADHD was quite common, yeah.
And I guess the second question builds on what you were saying. You know, this complicated interaction between genetic familial and environmental. Do you have measures of kind of caregiving style or anything to do with the caregiver parents in terms of how they interact with their offspring, and how that relates to the disorders?
Yeah, we have the parental bonding instrument. Yeah, which is really great, and yeah, you can see how the child, yeah.
Because I'm just wondering, you're in a great position now to be able to say, how does the parenting style actually affect the development of these other disorders that in turn perhaps are precursors to bipolar disorder? Is it nothing to do with caregiving environment, everything to do with genetic risk, or is it to do with the caregiving environment? You know, that's what would be so fascinating.
Yeah, and it's really amazing because we have genetic data of both parents and of the children. At least, well, we're collecting it. So yeah, that's something that. So I just started my PhD a year ago, so that's something that I will look into for the coming years. To think, how does this genetic and environmental factors interplay? Yeah.
Dankeschön.
That's German. In Dutch, you say dankjewel. Yeah.
Great presentation. It's awesome to hear the genetics on so lots of those papers. Two simple questions. One is, do you have a cardiovascular risk factor picture of what
Yeah.
The first question was, do you have any cardiovascular measures?
Yeah, we have BMI, weight, like we also measure the hip and waist ratio, and blood pressure, yeah.
bias. The other question is, and I don't know if you're directly involved in this, but we're really interested in patient and family engagement. I'm wondering if you know at all what the nature of the discussions were with the youth panel and the adult panel, or how that structure works.
Yeah, so I wasn't involved back then, but I know more or less that oh, sorry. So the question was how the adult and youth panel was involved in the study. Yeah, so they were, for example, the part that I just mentioned, the screening, validation of the screening instrument, they were really closely involved in the wording of the questions, like, is this clear to you? Yeah, that's one example, what they did. And yeah, also just in a general setup, like, what do you think is important? And I think also we focus a lot on resilience. And I think that's also something that came from the youth and adult, also from the researchers, but also they thought it's also important to know, yeah, what protects this high risk offspring from developing psychopathology. Yeah.
Could you go back to the slide with the Achenbach findings?
Mm-hmm. This one.
And how well do you understand the Achenbach? I want to know if this is a fair question or not.
Well, just shoot and I'll see.
So these are the most widely used scales in international research. They've been translated into more than 50 languages. The Netherlands is one of the countries that has its own set of norms. But for most of the countries, Achenbach has said, I am the keeper of the norms. And so there's an international set. Do you know where these scores, a T-score of 50 is average compared to other people of the same biological sex and roughly age group. So is this average for the Dutch population or for the global?
Well, I calculated these T-scores and I just used the one that was on the questionnaire, that was in English. So I'm guessing that was,
it would be
not
the
in Dutch.
Achenbach.
Yeah.
So that makes sense to me. That's really, and so
What?
that's cool.
Okay.
So and you knew the answer. So particularly as you look at these, what this is saying is average in our sample is healthier than average compared to the norms. Average would be right at a 50. So one thing that we see a lot in studies is super controls where professors bring their kids and so you have an upper socioeconomic status so that would explain the blue bars being low. But the high risk groups also were low. So I looked at it and I was like, oh, that's something. But when you look across planet Earth and Achenbach says, whoa, I have all the norms. He says it's basically all the same, but there are three flavors. They're the average countries, the fewer problems than average countries, and the higher problems than average countries. So what would you guess about the Netherlands? Are they sort of globally average in terms of behavior problems, or lower than average or higher than average? Lower, they're lower. So this is like we're doing science or something. We're using a scale, comparing it to international norms because you did it.
Apparently,
yeah.
yeah.
So that's really, really, really neat. So I've got good news. I have a funder who would like me to ask you, but because you're in Europe, it's the €1 million question.
Okay, yeah. Well, I think the most important thing, so we now have funding to do follow-up assessment for 3 years. And definitely, I think, also because you see in this slide actually that apparently the psychopath, well, we kind of expected that increases, like the difference increase. So I'm very curious to see how this will go in like 10 years.
Excellent.
So I don't need $1 million. I also need a lot of time, I think. But and something else, so I'm interested in genetics and environment. So another like logical thing would be epigenetics as well.
Excellent.
Yeah.
Other questions? Somebody, could you like poke me? Because all the speakers ended on time. I don't think this is real, but apparently it is. So well done.
Okay. Thank you.
So thank you very much to all of our presenters and to Mary and to Eric for doing a great job hosting these sessions, which I thought were really engaging. Eric, the $1 million question, did a great job, and then Mary, the panel was amazing. So thanks so much. This brings us to the conclusion of our day. So the first thing I wanted to do is just thank everybody for sticking with us, for participating. I thought there's a lot of thought-provoking questions, which is exactly why people, especially early career, are presenting to get them thinking. And I think that there was a broad spectrum of those questions which served that task. And for those who've stuck it out online, thanks very much, and consider coming in next year in person. As a next step, I wanted to.
One thing which I forgot to say.
Go ahead.
Sorry, this happens as I get older. I'm like, oh, wait, one more. I also want to give a shout out to all three panelists for wonderful, wonderful presentations, but also to their mentors because they were extremely well prepared, did a wonderful job with the presentations. And those were very sophisticated questions and very thought-provoking, like you said. And you guys were ready. That's really, really cool.
So we have two poster presentation winners. So we had judges, as you know, and thanks for the judges who all spent the time to go ask questions and assess the science and the presentation. And we have a second place winner and a first place winner. So our second place poster winner is Emily House-Vallis. So congrats, Emily, come on up.
Picture.
Which was a flag.
Thank you.
And our first place winner is Dana El-Soufi El-Sabag, if I pronounced her name correctly. And then the one final comment is for those of you who still have energy, we have a great patient and family public information night that starts, doors open at 6 p.m. tonight. It's 2 flights of stairs up from here. We hope to have a youth presenter, hear from our CEO, hear from 3 of our early career investigators on the topics of cannabis, vascular, and DBT in youth with bipolar disorder. I'll present, and we'll also hear from a lived experience perspective from the Mood Disorders Association of Ontario. So if you have energy, stick around, let us know if you want to stay. And thank you very much for being here today.